Welcome to our deep dive on cell theory.
Oh, yeah.
We're going to be exploring a cell theory lesson today.
Okay.
And I'm really excited to unpack
Yeah.
how these tiny units of life revolutionized our understanding of, well, everything.
It's a journey.
Yeah.
It's a journey of discovery that spans centuries,
Right.
starting from the very first time someone peeked at a cell under a microscope,
Wow.
all the way to,
Yeah.
debunking some long-held beliefs
Right.
about where life comes from.
I've always taken it for granted that we're made of cells,
Sure.
but I've never really thought about how we even figured that out in the first place.
Right.
So let's start with the basics.
Okay.
What exactly are cells?
Cells are the fundamental units of life.
Okay.
Think about it like this.
Mm-hmm.
Every living thing,
Yeah.
whether it's a towering redwood tree
Wow.
or a microscopic bacterium,
Okay.
is built from these tiny building blocks.
Tiny building block.
Yeah.
I like that.
And cell theory basically lays out the ground rules.
Okay, ground rules. I like that.
Yeah, for how these building blocks work.
So what are the main principles of cell theory?
Cell theory states that all living organisms are composed of one or more cells.
Okay.
For starters,
Okay.
that makes cells the basic unit of structure and organization in all living things.
Got it.
And this is a big one.
Okay.
All cells arise from pre-existing cells.
Okay.
They don't just appear out of thin air.
Wait, so people used to think that life could just spontaneously pop into existence.
Exactly.
Like imagine finding a loaf of bread and suddenly, bam, mold. That's what they thought was happening.
It seems wild to us now.
Right.
But that idea,
Yeah.
called spontaneous generation,
Spontaneous generation.
Yeah.
Okay.
was actually a widely held belief for a long time.
Wow.
But thankfully, we'll get to the scientists who proved that idea wrong a bit later.
Okay, good.
First, let's step back in time to 1665,
Okay.
when an English scientist named Robert Hooke made a groundbreaking observation.
1665.
Wow, we're going way back.
Hooke was looking at a thin slice of cork under an early microscope,
Okay.
and he saw these tiny compartments that reminded him of the small rooms that monks lived in,
Uh-huh.
which were called cells.
Oh,
And that's how the term cell was born.
that's amazing. So just from looking at a piece of cork, he sparked a whole scientific revolution.
It's pretty incredible,
yeah.
right? It's like the foundation of all biology rests on looking at a piece of cork.
Right.
Then in 1674,
Okay.
Anton van Leeuwenhoek took things a step further.
Okay.
He observed living cells
Oh, wow.
in pond water, of all places.
Pond water. Okay, that's even wilder than cork.
Right.
So we went from cork to pond scum.
Yeah.
What happened next in our cell theory saga?
Our cell theory lesson takes us to the 1830s,
Okay.
where two scientists,
Okay.
Matthias Schleiden
Okay.
and Theodor Schwann,
Okay.
were hard at work,
Okay.
studying plants and animals respectively.
They both came to the same remarkable conclusion.
Okay.
All plants and the animals
Wow.
are made of cells.
Wow. That must have been a huge realization.
Yeah.
Suddenly, cells weren't just these curious little compartments. They were the fundamental building blocks of all life.
Exactly.
Wow.
It was a paradigm shift in how we understood the living world.
That's amazing.
And then in 1855,
Okay.
Rudolf Virchow entered the scene
Okay.
and proposed a truly revolutionary idea.
Okay.
He said that cells can only come from other cells,
Okay.
directly challenging the idea of spontaneous generation.
Okay, so this spontaneous generation debate is really heating up. We'll have to hear more about that later.
We will.
But first, I'm curious about the different types of cells.
Okay.
I remember learning about prokaryotes and eukaryotes,
Mm-hmm.
but I could use a refresher.
You're right. Not all cells are created equal.
Okay.
Prokaryotes are like the ancient ancestors of all cells.
Okay.
They're single-celled organisms.
Okay.
And they're pretty simple compared to eukaryotes.
Okay.
They don't have a nucleus or other membrane-bound organelles.
So prokaryotes are like the minimalist version of a cell.
Exactly.
Okay.
Bacteria and archaea fall into
Right.
this category.
Okay.
They've been around for billions of years,
Wow.
and they're incredibly diverse and resilient.
That's amazing.
Now, eukaryotes, on the other hand,
Okay.
are more complex. They
Okay.
have a nucleus,
Right?
which contains their DNA,
Uh-huh.
and other membrane-bound organelles that act like specialized compartments within the cell.
Okay, so eukaryotes are like the upgraded deluxe model with all the bells and whistles.
A good way to think about it.
Okay, cool.
Plant and animal cells,
Yeah,
including the ones that make up you and me,
right.
are all eukaryotic cells.
So we're talking collections of eukaryotic cells.
That's right.
That's pretty wild. I'm also remembering diagrams from textbooks showing those classic plant and animal cell shapes.
Mm-hmm.
The rectangular plant cells.
Yeah.
And the roundish animal cells.
You're thinking of some of the key differences in their structure.
Okay.
Those rigid rectangular shapes come from the cell walls that plant cells have for structural support.
Right, so they need those to
Yeah.
be strong.
And of course, plant cells have those chloroplasts
Right,
that allow them to do photosynthesis,
right.
using sunlight to make their own food.
Photosynthesis, the ultimate life hack.
It is.
So what about animal cells? What
Okay.
makes them different?
Animal cells don't have those rigid cell walls.
Okay.
They're more flexible
Yeah.
and can take on different shapes.
Right.
They still have a cell membrane that acts like a boundary,
Uh-huh.
but without the extra reinforcement of a cell wall.
Gotcha. Okay.
Yeah.
And what about the other parts inside, like the nucleus,
Okay.
mitochondria, and those storage units, the vacuum
You hit
storage?
all the key players.
Sweet.
The nucleus is like the cell's control center.
Okay.
It contains the DNA,
Yeah.
which acts as a blueprint for building and running the cell.
Mm-hmm.
Mitochondria are the powerhouses of the cell,
Okay.
converting energy from food into a form the cell can use.
Right, the energy factories.
Exactly.
And vacuoles are like the cell's storage units, holding water,
Mhm.
nutrients, and even waste products.
That's right.
It's incredible how each cell is like a tiny self-contained city
It is.
with all these specialized structures working together.
Yeah.
But if cells are the fundamental units of life,
Mhm.
how do they make sure life continues? I'm guessing it has something to do with making more cells.
You're exactly right. Cells need to divide and make more cells for growth, repair,
Yeah.
and reproduction of organisms.
That makes sense.
Yeah.
But how does a single cell actually divide and make a copy of itself? Is it just like splitting in two?
It's a bit more complicated than that.
Okay.
There are two main types of cell division,
Mm-hmm.
mitosis and meiosis.
Okay.
Mitosis is like creating a perfect clone. It produces two identical daughter cells,
Okay.
which is essential for growth and repair.
Okay.
So if you get cut,
Yeah.
mitosis is how your body replaces those damaged skin cells.
So mitosis is like hitting copy and paste on a cell. What about meiosis?
Meiosis is a bit more specialized.
Okay.
It's all about creating diversity.
Okay.
Meiosis produces sex cells, or gametes,
Uh-huh.
which have half the genetic material of a regular cell.
Half the genetic material. Why is that important?
It's all about creating unique combinations.
Okay.
When a sperm and egg cell combine during fertilization,
Right.
they each bring their half set of chromosomes,
Okay.
creating a new individual with a full set of genetic information,
Gotcha.
a mix of traits from both parents.
Ah, that's why meiosis is so important for evolution. It's constantly shuffling the genetic deck, creating new combinations and variations in each generation.
Exactly.
Wow.
It's like nature's way of experimenting with different combinations,
Right.
which drives the evolution of new species over time.
Yeah, that's amazing.
But before we go down that evolutionary path,
Okay.
I think it's time to circle back to that spontaneous generation debate we left hanging.
You're right. I'm dying to hear how that myth was busted. So remind me, people actually used to believe that living things could just appear out of nowhere.
It sounds crazy now,
I know.
but that was the prevailing thought for centuries.
Real.
Think about it.
Yeah.
Without microscopes to reveal the tiny world of microorganisms,
Right.
people saw things like maggots appearing on meat
Yeah.
and assumed they just spontaneously arose from the meat itself.
Oh, gross.
Even Aristotle, the famous Greek philosopher, proposed this idea.
I guess it's a classic case of seeing is believing
Right.
without the tools to see the unseen.
Yeah.
It's easy to jump to conclusions.
Precisely.
Yeah.
But thankfully, science is all about questioning assumptions
Right.
and digging deeper.
Yeah.
And that's where our scientific heroes come in.
Okay.
Scientists like Francesco Redi,
Okay.
Louis Pasteur, who designed clever experiments
Okay.
that challenged this long-held belief.
Okay. I'm on the edge of my seat. Tell me more about these experiments.
Let's start with Francesco Redi.
Okay.
Back in the 1600s,
Okay.
he conducted a simple yet elegant experiment using jars of meat.
Oh, okay. I'm picturing this.
Some jars were left open,
Okay.
some were sealed tightly,
Gotcha.
and others were covered with gauze.
Okay. What happened? Did maggots just magically appear in all the jars?
Nope.
Okay.
Redi observed that maggots only appeared in the jars that flies could access the open jars,
Oh.
the sealed jars,
Right.
and the ones covered with gauze,
Okay.
which prevented flies from laying their eggs on the meat,
Right.
remained maggot-free.
So it wasn't the meat itself creating the maggots.
Right.
It was the flies.
Exactly.
That's a pretty convincing blow to spontaneous generation.
It was a major step forward.
Okay.
But the debate wasn't quite over.
Go.
Fast forward to the 1800s.
Okay.
And enter Louis Pasteur, a French chemist and microbiologist.
Okay.
He used a special type of flask called a swan-necked flask to conduct his experiments.
Swan-necked flask. Okay, I'm intrigued.
Yeah.
What's so special about that?
These flasks have a long curved neck
Okay.
that allows air to enter,
Okay.
but prevents dust and microorganisms from falling directly into the broth inside.
Oh.
Pasteur sterilized the broth in the flasks,
Okay.
killing any existing microorganisms,
Okay.
and then observed what happened over time.
So did anything grow in the sterilized broth?
In the swan-necked flasks,
Wait.
the broth remained clear,
Okay.
free of any microbial growth.
Wow.
But when he tilted the flasks,
Okay.
allowing the broth to touch the dust and microorganisms trapped in the curve of the neck,
Oh.
the broth quickly became cloudy with microbial life.
Yeah, huh. So even with access to air, life didn't just spontaneously appear in the broth.
Exactly.
It had to be introduced from an external source.
Precisely. Pasteur's experiments were a resounding victory for the idea that life comes from pre-existing life.
What a mic drop moment for science. But I'm curious, where does Rudolf Virchow fit into all of this?
Okay.
He's the one who famously said all cells come from cells, right?
You got it.
Okay.
Virchow's work,
Mm-hmm.
building on the findings of René Pasteur
Yeah.
and others,
Right.
solidified the concept that life doesn't magically appear.
Right.
It passed down through generations of cells.
It's like a continuous chain of life,
It is.
stretching back billions of years.
Exactly.
That's an incredible thought.
It is.
Yeah.
It really is mind-blowing when you think about it that way, isn't it? And it raises even more questions.
Yeah.
I mean, if all cells come from other cells,
Right.
how do we get so much diversity?
Yeah.
Like, how did we go from single-celled organisms
Right.
to all the complex life we see on Earth today?
That's a great question. And it leads right into the next thing I was wondering about. How do cells even become different from each other in the first place?
Yeah.
They all start from the same basic blueprint, right?
That's a question that still fascinates scientists today.
Really?
It's a process called cell differentiation.
Cell differentiation.
Yeah, and it's all about how cells specialize
Okay.
to perform specific functions.
Okay, so walk me through this.
Okay.
How does a cell decide to become a brain cell versus a muscle cell, for example?
Imagine a stem cell as a blank slate.
Okay.
It has the potential to become any type of cell in the body
Wow.
during development.
Okay.
These stem cells receive signals
Okay.
that guide them down a particular path,
Okay.
influencing which genes are turned on or off.
So it's like a cellular choose-your-own-adventure story.
Exactly.
What kind of signals are we talking about?
These signals can come from neighboring cells,
Okay.
the environment,
Okay.
even the cell's own internal programming.
Wow.
It's a complex interplay of factors
Yeah.
that scientists are still unraveling.
It's like a secret language between cells.
Yeah.
Guiding them to their destinies. But I have to wonder, if we can understand this language, could we manipulate it? Could we tell a stem cell to become whatever type of cell we need?
That's one of the most exciting areas of research right now.
Really?
In regenerative medicine,
Okay.
scientists are working on techniques to coax stem cells into becoming specific cell types
Okay.
that could be used to repair damaged tissues or even grow new organs.
That's incredible. We could potentially cure diseases and extend human lifespans
Yeah.
through the power of cells. Are we talking about science fiction here?
I know.
Or is this really happening?
The possibilities are vast, and the research is advancing rapidly.
Wow.
While we're not quite at the point of growing entire organs in a lab,
Okay.
we're making significant progress.
That's great.
For example, scientists have been able to create insulin-producing cells from stem cells,
Wow.
offering hope for people with diabetes.
It's amazing to think about how much potential there is in these tiny building blocks of life.
It is.
But it also makes me think about the bigger picture.
Okay.
We've talked about cells coming from other cells, but how did the very first cells even arise?
That's one of the biggest mysteries in science.
Yeah.
The origin of life
Yeah.
is still a topic of intense research and debate.
Really?
There are many hypotheses, but no definitive answers yet.
So no one knows for sure how that first spark of life happened?
Not yet.
Oh, wow.
But scientists are piecing together clues from various fields.
Okay.
like chemistry, geology, and biology,
Okay.
to try to understand the conditions that might have led to the formation of the first cells.
Okay, so maybe we don't know how the first cells appeared,
Right.
but we do know that once they existed, they began to evolve and diversify, right? I mean, look at the incredible variety of life on Earth.
You're absolutely right.
It's incredible.
The evolution of life is a testament to the power of cells
Yeah.
to adapt and change over time.
Right.
And the key to understanding this is in those tiny changes
Okay.
that happen within cells themselves.
So you're talking about mutations, those changes in the genetic code that we hear about.
Exactly.
Okay.
Mutations are like typos in the DNA,
Okay.
and they happen all the time during cell division.
Right.
Most mutations are harmless or even detrimental,
Okay.
but occasionally a mutation arises that gives a cell a slight advantage in its environment.
So it's like natural selection playing out at the cellular level.
Precisely.
Okay.
Cells with beneficial mutations are more likely to survive and reproduce,
Right.
passing those advantageous traits onto their offspring.
That makes sense.
And over vast stretches of time,
Yeah.
these tiny changes can accumulate,
Right.
leading to the evolution of new species.
It's mind-boggling to think that all the diversity of life we see around us stems from these tiny changes in cells.
It is.
Over millions and billions of years.
It really puts our own existence into perspective, doesn't it?
It does.
We're part of this incredible lineage of life
Yeah.
that stretches back to those very first cells.
It makes me look at a blade of grass or a buzzing bee in a whole new light.
Yeah,
We all share a common ancestry at the cellular level.
we do.
But speaking of perspectives,
Okay.
our deep dive into cell theory wouldn't be complete without considering the ethical implications of all this knowledge, right?
You're bringing up a crucial point.
Yeah.
As our understanding of cells deepens,
Right.
so too does our ability to manipulate them.
Right.
And with this power
Yeah.
comes a responsibility
Yeah.
to consider the ethical implications
Yeah, that's a good point.
of our actions.
So where do we draw the line?
Right.
How do we ensure that our exploration of the cellular world benefits humanity without crossing any ethical boundaries?
Those are questions that scientists, ethicists, policymakers, and the public
Wow, so everyone needs to be involved.
need to grapple with.
Yeah.
We need to have open and honest conversations about the potential benefits and risks
Right.
of these advancements.
I'm ready to dive into those ethical considerations. This is where things start to get really interesting, in my opinion.
I agree. Let's explore that fascinating territory in our final segment.
We've been on quite a journey today, haven't we?
We have.
From the very first glimpse of a cell under a microscope
Great.
to the realization that we're all connected through this intricate web of cellular life.
It is an intricate web.
Yeah.
Yeah.
But as we've hinted at, this knowledge also comes with some big questions about how we use it.
Big questions, yeah.
You left us hanging before with
Right.
that idea of responsibility.
Okay, sure.
What are some of the specific ethical considerations we should be thinking about
Okay.
as our understanding of cells grows?
Let's start with genetic engineering.
Okay.
As we unlock the secrets of the genes that control cell function,
Right.
we're gaining the ability to modify those genes.
Right.
That opens up incredible possibilities for treating diseases.
Right.
Things like gene therapy to correct genetic defects, or even potentially engineering cells to be resistant to certain viruses.
Wow.
But it also raises some tough questions about where we draw the line.
I can see how that could get tricky.
Yeah,
What starts as an effort to cure diseases
right.
could potentially lead to trying to enhance human capabilities.
Right.
Where do we draw the line between therapy and enhancement?
That's the million-dollar question.
Yeah.
And it's one that society needs to discuss and debate.
Yeah, definitely.
Who gets to decide what constitutes an enhancement?
Right.
And what are the potential consequences of altering the human genome?
Yeah.
Not just for individuals,
Right.
but for future generations.
It's like we're stepping into uncharted territory.
Yeah,
And we need to proceed with caution.
we do.
Are there other areas where the ethics of cell manipulation become especially important?
Absolutely.
Okay.
Cloning is another area that raises profound ethical questions.
Right.
While there might be potential benefits in agriculture or medicine,
Mm-hmm.
the idea of creating genetically identical copies of organisms
Yeah.
raises concerns about the uniqueness of individuals
Right.
and the potential for exploitation.
It's like something out of a science fiction movie.
It is.
But these are real issues we're facing.
They are.
It seems like stem cell research also falls into this category of ethically complex territory.
You're right.
Yeah.
Stem cells hold immense promise for regenerative medicine,
Right.
with the potential to repair damaged tissues and even grow new organs.
Yeah, it's amazing.
But the source of those stem cells,
Right.
especially embryonic stem cells,
Okay.
has been a subject of ongoing ethical debate.
So how do we navigate this complex ethical landscape? Seems like every step forward in our understanding of cells brings a new set of ethical challenges.
It does, it's true.
Yeah.
I think the key is open and honest dialogue.
Okay.
We need to bring together scientists, ethicists, policymakers, and the public.
So a big group.
Yeah, a big group. To discuss these issues and develop guidelines
Right.
for responsible innovation.
Yeah,
We need to weigh the potential benefits of these technologies against the risks
definitely.
and make sure that their development and use align with our values as a society.
It's a lot to consider.
It is.
But it's clear that we can't shy away from these ethical conversations.
Yeah, we can't.
They're crucial to ensuring that our exploration of the cellular world
Right.
benefits humanity in a responsible and ethical way.
Absolutely.
This deep dive has been an incredible journey.
Yeah,
A journey into the microscopic world of cells
yeah.
and a journey into the complex ethical landscape
Right.
that comes with understanding these building blocks of life.
It's a journey that's still unfolding,
Yeah,
and it's one that we're all taking together.
that's a nice thought.
Yeah.
Thanks for joining us on this deep dive into the fascinating and thought-provoking world of cell theory.
Yeah.
We hope you've come away with a newfound appreciation for the tiny wonders that make life possible,
That's right.
and a sense of the responsibility we all share as we continue to unlock the secrets of these amazing cells.
Yes.